Welcome back to Microbiology. We're going to continue with Chapter 5. For the third video, our focus is going to be fungi. By the end of this video, you should be able to explain why we study fungi in microbiology, describe their unique characteristics, compare how fungi reproduce both sexually and asexually, identify major groups of fungi and some examples of each, and recognize medically important fungi that cause disease or produce toxins, and finally, classify fungal organisms into their proper groups. So why do we study fungi in microbiology? Fungi have macroscopic large forms like mushrooms, but they also include microscopic forms that we can't see with the naked eye, like yeast, molds, and spores. Some fungi cause mycoses or fungal infections, and mycoses is plural. If it was singular, we would say mycosis with an IS. But many of these are also opportunistic, affecting people with weakened or compromised immune systems. Fungi also play many important beneficial roles in our lives, including acting as decomposers so they can recycle nutrients in our ecosystems. And they're also important in food production in things like bread, beer, or cheese. Finally, we also have some that are sources of medications, like the very well-known antibiotic penicillin. Fungi are eukaryotic and heterotrophic, meaning they cannot make their own food. They have to get their nutrients from different organisms. And they're also saprozoic, and this means that they absorb dissolved organic material and salts from dead or decaying organic matter in their environment. Their cell walls contain chitin, unlike plants. Plants use cellulose in their cell walls. And their membranes, the fungal cell membranes, contain ergosterol instead of cholesterol, what we have in human cell membranes. So this makes this ergosterol an important drug target for antifungal medications, as you can target ergosterol, and this allows the antimicrobial drug to kill fungi without harming human cells, right? We don't have any ergosterol. We have cholesterol instead. In terms of the forms of fungi that exist, we have molds. Molds are multicellular and grow as hyphae, these long filament structures. When they're clumped together, they form a structure known as a mycelium. And we also have yeasts. Yeasts are unicellular and they reproduce by budding. Finally, we have some fungi that are dimorphic, and this means that they can actually switch between yeast and mold forms depending on their environment. And here I just wanted to show you a picture of how different molds and yeasts look from one another. We have molds here on the left. They're multicellular and they're filamentous, so you can see these string-like structures, and many of them reproduce through spores. So I can see a few spores here that can be released into the air, land somewhere else, and produce more mold. On the right here, I have yeasts. You can see these are single-celled organisms, kind of like an egg-shaped cell right here, and they reproduce by budding. This figure shows different forms of fungi. For molds, shown here at the bottom left, these two, these form hyphae. So molds form hyphae, those filament-like structures that can be septate or non-septate. For septate hyphae, these have walls between the cells. So I can see a distinct line separating each cell from the next. Hyphae that lack walls are shown in the middle here. They lack cell walls and lack cell membranes between the cells. So it looks like just one long continuous structure. These are non-septate, also known as cenocytic. Hyphae, these hyphae can form a tangled network. When they're all bunched together, they're called a mycelium or mycelia, plural. And they can form the thallus or body of fleshy fungi. So up here, what we recognize usually as mushrooms. Down here on the bottom right, we have yeasts. Earlier, I showed you some picture of oval-shaped cells. They're unicellular, but sometimes they can form pseudohyphae. So budding yeasts reproduce asexually by budding off a smaller daughter cell, but sometimes these cells stick together and they form that short chain or pseudohyphae. The structural differences between these different types of fungi are really key for their identification. I mentioned dimorphic fungi earlier. These are very interesting because they can alternate between mold and yeast form depending on their environment. Many are important medically. So for example, we have Histoplasma capsulatum here. This causes a lung disease called histoplasmosis, and it's found in soil that's been contaminated with bird or bat droppings. Candida albicans, a common part of our normal microbiota, that can cause yeast infections, oral thrush, and skin infections when it overgrows, is also a type of dimorphic fungus. These organisms, so dimorphic fungi, can switch forms, these two in particular, depending on the temperature of the environment. So they grow as molds at room temperature. So usually room temperature or ambient temperature is close to 25 degrees Celsius. But inside the body, where we have about 37 degrees Celsius, they grow as yeasts. And this flexibility really increases their ability to infect humans. This image shows Histoplasma capsulatum. So outdoors, it grows as a mold. But once it's inhaled into the lungs, it converts to a yeast form where it can better multiply and cause disease. So this is why activities like cave exploring or spelunking can put people at risk. So this is just for fun, but spelunking involves exploring wild cave systems. It's also sometimes known as caving. It involves walking through the caves, crawling, or climbing or rappelling into the caves. And this is a picture of me from a long time ago caving. This was the biggest room that we found during our 4-hour trip. So we were underground for about 4 hours, and it's pitch black. It looks bright here because I asked my husband to take a picture of me, and we had to go through many, many tiny little openings like this where we could barely squeeze through. And I wanted to show you this because along the way, you may see many, many bats, as we did. We saw many bats. You have to make sure you don't disturb them. You don't want to get scratched or bitten, and you don't want to, or you want to be careful that you're not exposed to potential Histoplasma capsulatum that could be in their feces. So when we first got there, I didn't have a picture of us doing it, but we had to rappel down into the cave. And this is us at the end. So me and my husband, our outfits were new when we got in. We just bought some stuff from Walmart before the trip. And then you can see our knee pads. For me, I could squeeze through many of the small openings, but I really tore up my knee pads, whereas my husband, he could barely fit through some of the openings. So you could see a lot of dirt on his shirt because he had to really push through those openings. So my husband planned this trip, and when he was describing it, I was imagining something like this in my mind. In reality, it turned out to be like this for pretty much the whole 4 hours. It was still a ton of fun in the end, and I enjoyed it, but if you ever plan or think about going spelunking, just make sure you do your research. A lot of people get down there. It's extremely crawling through tight spaces. It's pitch black if you lose or your battery runs out in your headlamp. So just be careful. Make sure you're okay with small openings. Watch out for, make sure you're not going if it's raining. There could be floods down there that could lead to drowning. There could be collapse of the caves. So you want to go somewhere reputable. Watch out for head injuries. Watch out for bats, right? And make sure you have people who are quite experienced leading the trip. All right, back to fungi. So fungi can reproduce in two main ways, asexually or sexually. In asexual reproduction, this can occur through budding, fragmentation, or by forming asexual spores through mitosis. For sexual reproduction, some fungi can produce gametes of two mating types. These gametes will undergo plasmogamy, where cytoplasms fuse together, and then karyogamy, where their nuclei then fuse, and this will form a diploid zygote. This then undergoes meiosis, producing sexual spores, such as zygospores, ascospores, or basidiospores, depending on the fungal group. Our book gives us a nice example of reproduction of fungi, where this life cycle shows both asexual and sexual reproduction stages. Don't worry about these terms, any terms that I don't mention. I just want to go over the general process. So starting at the top here, we have asexual reproduction. Right here we have haploid spores, similar to human sperm and egg, except that these spores are able to undergo mitosis, forming copies of themselves. So this is asexual reproduction. They're dividing and they're forming these filamentous structures, these hyphae. Remember the hyphae are like the string-like structures. These look like septate hyphae, right? They have cell walls in between each cell. A cluster or mass of hyphae is a mycelium. Here they've put the plural form mycelia here. And I see two different types of cells or gametes here. Looks like some of them have these red nuclei and the others have green nuclei. In plasmogamy, the cytoplasms are going to fuse. So they're trying to show some fusion, and by the time we get to here, we can see that each of these cells have both nuclei, right? Within the septate hyphae, the individual cells, I see both red and green nuclei. Then in karyogamy, I can see that the red and green nuclei have fused together to form a diploid zygote, right? Kind of like what happens when sperm and egg come together. You form a diploid organism. The first cell is known as the diploid zygote. These then undergo meiosis, and we're going to reform more haploid gametes. So gametes are going to be formed. These are haploid, and then they're going to make copies of themselves through mitosis, forming more spores and restarting the cycle. Here I just wanted to show you a few examples of molds and their structures. Here on the left we see more hyphae, those filament-like structures. At the end of the hyphae in this particular mold, we have a sac of spores. So this structure is known as a sporangium. If the sac breaks open, the spores get released. Bread mold, or rhizopus, has something similar. We have a bunch of hyphae, those string-like structures, forming this mass, a mycelium. If you were to zoom in, you can kind of see these darker dots. Those are also sporangium, full of spores. Here's our first checkpoint. So is a dimorphic fungus a yeast or a mold? Remember that a dimorphic fungus can be either, right? Either a yeast or a mold. Really depends on their environmental conditions. For an example, we talked about Histoplasma capsulatum that grows as a mold at 25 degrees Celsius at ambient room temperature or in the environment, but it switches to a yeast form at 37 degrees Celsius, right? The temperature of the human body. And this ability really gives them the ability to live in diverse environments and contributes to their infectivity inside our bodies. There are four main fungal groups of medical importance, and the first two are shown here. This is phylum Zygomycota, like Rhizopus, which is also a common bread mold. These are mostly saprophytes, so they feed on dead organisms, but some can cause opportunistic infections in immunocompromised individuals. The next phylum, Ascomycota, is actually the largest group and includes several examples here. We have Aspergillus, different species of Aspergillus, which can sometimes be found in contaminated nuts or stored grains. This is linked to allergies, infections, and potential aflatoxin contamination, which is a toxin and a carcinogen. Penicillium is the source of penicillin, the antibiotic. Dermatophytes, such as trichophyton and microsporum, can cause skin infections like ringworm or athlete's foot. Dimorphic fungi, like Histoplasma, Blastomyces, and Coccidioides, these cause respiratory infections. We have Candida albicans, responsible for yeast infections, and then Saccharomyces, such as Saccharomyces cerevisiae or baker's yeast, can be used in baking and brewing. Here we have another picture contrasting molds and yeast. We have mold here in the middle, Aspergillus. This looks like part of a hypha. this filament-like structure with spores at the end. And then we have our single-celled yeast, Candida, which causes yeast infections or thrush. And our two other important groups, so this is continued, are Basidiomycota and Microsporidia. For the first one, Basidiomycota, these include mushrooms and puffballs, and also several important or notable examples like Cryptococcus neoformans. This causes lung infections in immunocompromised patients. And Amanita phalloides, also known as the deadly death cap mushroom, produces toxins that block transcription, so making us unable to produce messenger RNA from DNA. This one is shown here on the left, and it's within the same genus as a very famous mushroom, Amanita muscaria. This is probably one of the most recognizable fungi in the world because of its appearance in different TV shows or children's stories or video games. And then Microsporidia down here, these are obligate intracellular parasites because they don't have several organelles like mitochondria. So one example is enterocytozoan Vianusi, and this can cause diarrhea and gallbladder disease. I believe it's misspelled in the book. I think they accidentally put A in there for their textbook. Our next checkpoint asks, which group of fungi appears to be associated with the greatest number of human diseases? This was or were the Ascomycota. So these fungi in the phylum Ascomycota, including pathogens such as Candida albicans, Aspergillus. We had those dermatophytes that cause the skin infections, and our dimorphic fungi like Histoplasma, Coccidioides, or Blastomyces. It turns out that treating fungal infections is more challenging than treating bacterial infections, and that's because fungi, like us, are eukaryotic. This means that their ribosomes, their cytoskeleton, their cell membranes are similar to ours. So far, the main antifungal drug targets are ergosterol synthesis, targeted by a group of drugs known as azyl drugs, or the chitin cell wall. So what is ergosterol? Remember that in the cell membrane from Chapter 3, there are lipids embedded in the membrane for stability and fluidity. In humans, the sterol in our membranes are cholesterol molecules, cholesterol. But in fungi, they use ergosterol, which we do not have. So this is a nice target that's unique to fungi. Chitin cell walls, remember humans, we don't have cell walls, so targeting chitin is also specific to fungi. But systemic antifungal drugs often have a lot more toxic side effects than antibiotics, again because of our cellular similarities. Even though we're trying to kill fungi, we might accidentally hurt our own cells instead because of these similarities. All right, let's recap 5.3. So why do we study yeast and molds and fungi in micro? It's because many fungi, like yeasts, molds, and fungal spores, are microscopic. Fungal diseases, mycoses, are often diagnosed using microscopy. So their microscopic nature and medical importance really place them within our field of microbiology. For the second one, describe unique characteristics of fungi. They're eukaryotic, heterotrophic, and saprozoic. Remember, they absorb nutrients. They have cell walls made of chitin, not cellulose like plants do. And their membranes have ergosterol instead of cholesterol, what we have. And ergosterol can be a target for antifungal drugs. For their forms, remember they had the mold form with multicellular hyphae forming a mycelium. Or they could be yeasts, unicellular, that reproduce by budding. Or they could be dimorphic fungi that switch between molds and yeast, really depending on their environment. For the next one, examples of asexual and sexual reproduction. We talked about asexual reproduction, that could be mitosis through budding, or fragmentation of the hyphae, or by forming asexual spores. For sexual reproduction, I showed you that cycle where we had plasmogamy, where the two different gametes, we had green and red in that picture, fused together to form the cytoplasm, or they fuse their cytoplasm. and then karyogamy, where the nuclei fuse to form a diploid zygote. And through mitosis, they would produce spores. And the spores produced really depends on the type of the fungus. We can make zygospores or ascospores or basidiospores, and we'll see these more in lab in the middle of the semester. For the fourth one, comparing major groups, we talked about zygomycota, like bread molds, like Rhizopus. These are mostly saprophytes. Some of them are opportunistic. The second group, ascomycota, these were the largest group, which included fungi such as Candida albicans, Aspergillus, dermatophytes, or histoplasma. Our third group were the Basidiomycota, which included mushrooms and puffballs, and included also examples such as Cryptococcus neoformans that can cause lung infections, and Microsporidia. These were obligate intracellular parasites that lack a lot of normal organelles, and an example could be Enterocytozoan bianusi, which causes diarrhea and gallbladder inflammation. Some examples of primary causes of infection. So this kind of overlaps with the previous one. We had yeasts like Candida albicans, which can cause yeast infections or thrush. Cryptococcus neoformans is also a yeast, which can cause lung infections. For molds, we talked about Aspergillus. That causes, it could be an allergy, like an allergic reaction, or dermatophytes that cause things like athlete's foot or jock itch or ringworm. And we had dimorphic fungi like Histoplasma capsulatum that cause respiratory diseases. For toxin-producing fungi, we talked about Aspergillus. They can produce aflatoxin, and it's also a potential carcinogen. We often find this in contaminated grains or nuts. Or Amanita phalloides, that death cap mushroom, which can produce deadly toxins too. Those toxins affected transcription, right? The production of messenger RNA. And then for major groups, again, we have those four groups. Zygomycota for molds like Rhizopus. Ascomycota, our largest group, had yeasts, molds, and many pathogens like Candida, Aspergillus, Histoplasma, they're dermatophytes. Basidiomycota were our third group. These included mushrooms or puffballs or Cryptococcus and Amanita. And then Microsporidia, these were obligate intracellular parasitic fungi like the Enterocytozoan. All right, that's it for fungi. In our last video for chapter 5, we're going to look at algae and lichen.